I'm not on board with testing longevity drugs in humans. Will nicotinamide riboside extend human lifespan? There's not even a way to do that experiment. How would you do that experiment? I'm not fully convinced when people say NAD declines in aging. They said NMN enters cells immediately. If you've taken any high school chemistry, things don't happen immediately.
This is a conversation with Charles Brenner. He's a professor and the Alfred E. Mann Family Foundation Chair in Diabetes and Cancer Metabolism at the City of Hope, where his research focuses on NAD metabolism and its role in aging, stress, and disease. He's famous for being one of the most outspoken critics of hype in the longevity field, challenging bold claims about age reversal. This is a Acc Podcast. Please enjoy. Your research, primarily focuses on NAD metabolomics. And NAD is this really critical molecule in so many different processes, most famously glycolysis and respiration, but also DNA repair, stress response, and aging. It's pretty well known that NAD levels decline with age, and that decline has been linked to various age-related diseases. Your lab discovered a new precursor called NR, which is a precursor to NAD, and you found that when you supplemented yeast with NR, it extended their lifespan. How long was that measurable extension of lifespan? And how do you compare these results that you see in these model organisms like yeast, mice, to what you might actually expect in humans?
I don't compare yeast aging to humans, and I don't think people should compare yeast aging to humans. And in fact, the model of yeast aging that we used is a very unreasonable model of aging. So I want to step back and say, yeah, what we've done in the NAD field is substantive and foundational and important and underlies core metabolism in every living thing. But I don't think that it's straightforward to say that boosting NAD metabolism will extend lifespan in humans. So essentially what our work has shown is that the NAD system, as central as it is to metabolic processes, is disturbed in a lot of conditions of metabolic stress. For example, when DNA gets damaged by the sun or by reactive oxygen species, the NAD system gets disturbed. In neurodegeneration, both central neurodegeneration, like in Parkinson's, Alzheimer's, as well as in peripheral nerve damage, the NAD system gets disturbed. In infection and inflammation, the NAD system gets disturbed. And so we have found, basically from our work in model systems, a roadmap for finding in the human condition where the NAD system is likely to be disturbed. We can usually measure those effects. Like in human mitochondrial disease, the NAD system is disturbed. In human alcoholics, the liver NAD system is disturbed. In people that are infected with COVID and the flu, the NAD system is disturbed in inflammation. And clinical trials that are designed to measure anti-inflammatory activity work. There's now 8 of them, randomized clinical trials, in which nicotinamide riboside provided at a gram a day doses. You look at legitimate conditions in which the NAD system is disturbed, you can find use cases for nicotinamide riboside, and you get success in those trials. If you look at unreasonable things, like will nicotinamide riboside extend human lifespan, there's not even a way to do that experiment. How would you do that experiment? I don't know. You could enroll people that are 105 so that you could live long enough to do that experiment. But people who are 105 are not like most of us. They're better than most of us in some ways that we don't understand. And the problem, I think, that may plague part of the bioacceleration movement is, and I didn't know that's what the movement was called, but I've been doing that my whole adult life. I've been doing biomedical research to try to improve the human condition. If you predicate the goals of the bioacceleration movement on the results that are least likely to be reproducible, or on model systems that don't apply to humans, the bioacceleration movement is making very bad choices and very bad investments. So when you ask me to what degree yeast lifespan extension results can be translated to human, I'm honest with you and I tell you, not at all. If you want me to spend the time, I'll tell you why the yeast system in which NR is active is not a good model for aging. And most yeast people won't even take the time to explain that to you. But it's not a model for any animal aging. It's a very unique thing to yeast replicative longevity that doesn't even apply to the model of how long a yeast cell lives. It's a very unique. And the thing is, you have to do a deep dive into research to figure out whether it is reproducible, whether it's hyped, and whether it can apply to another system. And I think that's what my lab is known for, is that we do foundational work and we apply it to things that are real, and we try to avoid things that can't be reproduced or don't apply to the human condition.
You're definitely one of the strongest proponents, I think, in this field, that a lot of the studies that we're seeing coming out of like longevity or life extension labs, you know how long and how extensive the clinical trial pipeline is for the FDA. And preclinical is literally step zero. It's not even, and there's phase 1, 2, and 3. And so having preclinical data that shows potential efficacy is not enough to really make those bold claims. And to your point, these models are not necessarily, good models for studying what might work in humans. And we see so many times that the data, that works in mice, it completely fails in humans. And you made an interesting point. I mean, you've shown that NAD levels aren't just affected by age, right? They're disrupted in things like viral infections, diabetes, postpartum lactation, even brain injury. There's so many different scenarios where NAD is important. And so my question is why do NAD levels decrease with age? Is it a cause of aging? Is it a consequence of a cause that's still unknown to us? I'm sure this is a question that many people are trying to answer, including yourself, but the NAD system seems quite versatile in my view, just based on even research you've done. And so I'm curious about sort of the specificity of it in aging, which a lot of people say it is, but maybe you have different opinions about that.
So the first thing I would say is that I am far more certain about the NAD system being disturbed in conditions of metabolic stress than by the process of aging. I'm actually not fully convinced when people say NAD declines in aging. My next question is, in what tissue, in what system, and what are all the details, and how did you come to that conclusion? And sometimes it's not all that clear what they're measuring and how applicable it is across the same organism in which they're looking at. So, for example, there are people that have shown that the NAD system declines in the liver during mouse aging. But I think when you look in the muscle, you don't see a decline. And I want to, does it depend upon the microbiome? Does it depend upon the mouse strain background and so forth and so on? That's why I also personally don't have the patience to do experiments that at primary endpoint of aging in a mouse. Aging as fast as it is with respect to human aging is probably not a great model for human aging. Like older people can die from falls. That's not uncommon. Mice never die from falls. Mouse lifespan can be extended by rapamycin. I don't believe that human lifespan can be extended by rapamycin or any type of mTOR inhibitor. And humans do better by maintaining their skeletal muscle mass as we age and mTOR inhibitors are going to dampen the central regulator of skeletal muscle mass maintenance and expansion. The NAD system does get disturbed in many conditions of metabolic stress through a number of different mechanisms. For example, in peripheral nerves, let me just say what a peripheral nerve is. You can feel things all the way to your fingertips and to your toes, right? And in conditions of metabolic stress that include poor glycemic control, so diabetes, and chemotherapy, these peripheral nerves, which are literally the longest cells in a mammal's body, right? They're meters long in elephants and giraffes, and they're pretty long in humans. These very long cells have incredibly long axons that are transmitting information all the way to your fingertips. Axons can be damaged. When the axon is damaged, an NAD intermediate called NMN accumulates and activates an enzyme called SARM1, and then SARM1 turns NAD into cyclic ADP ribose. It releases calcium, and it can result in destruction of the axon and the nerve. NMN, intracellular NMN, which a lot of people take as a supplement, intracellular NMN accumulation in a peripheral nerve sets off a cascade that leads to destruction of NAD and can cause axonopathy and ultimately irreversible nerve damage. So that's one very specific mechanism. The target there, were what we would try to inhibit, is an enzyme called SARM1. If you could inhibit SARM1, you might allow people to survive chemotherapy or type 2 diabetes with their peripheral nerves intact. In other conditions, it's something called activation of PARP1. So PARP1 gets angry when there's DNA damage, and it creates a polymer of ADP ribose. And it's there to assemble a DNA repair complex, but it's disturbing the NAD system at the same time as disturbing glycolysis and energy generation. In reactive oxygen stress, there's a metabolite called NADPH, which is providing electrons for detoxification of free radicals. And that system churns NAD. So there's many different conditions of metabolic stress that disturb the NAD system through unique ways. So that's my question to the rest of the field. When they see an observation in which the NAD system is disturbed over time, was it metronomic, time chronological based thing, or was it a metabolic insult based attack on the NAD system that becomes in some way cumulative? I don't know. And anybody that is telling you that they know is not thinking about what they don't know. That's my point of view.
Yeah, certainly. I feel like there's so many confounding variables that could lead to NAD depletion. And I think no one has really cracked yet, like, what is the cause of aging? Maybe it's, you've talked about evolution a lot. There's no reason why we should be surviving this long from an evolutionary perspective. And modern medicine has kept us, this long from a genetic perspective. Your view is that there's no feasible way we should be able to live past 120 or whatever we're encoded by. And you mentioned like an interesting point about sort of off-target effects, right? And I think that is also a really big topic of discussion in the field. There's some crazy startups that are claiming, like there's one on the West Coast that their whole thesis is we're going to upregulate telomerase to try and prevent the shortening of telomeres, which is associated with aging, right? Because as you get older, these cells, the chromosomes shorten over time. But obviously, you must know that telomerase, it's a prevalent oncogene in many cancers. And so there's also other arguments about different types of methods that have been explored. So like DNA methylation clocks, regressing cells back to a pluripotent stem cell sort of nature, like that could be a cause, or that could be a concern for cancer, giving people cancer without really knowing it. What are your thoughts on that? I'm sure you have strong opinions about what people claim they can do. This field is so ubiquitous. Everyone wants to live longer. And so it's gained a lot of mainstream attention from people who aren't necessarily scientists or maybe don't know what telomerase is. So maybe you could speak to a bit of that.
Okay, so there's a lot to unpack there. So why do we age, and what is the point of aging, and how much can you expect to extend lifespan beyond reproductive maturity? There is a driving force of what any animal can be encoded to do relates to reproduction and survival, right? Animals evolved in order to have some combination of fertility and fecundity and survival of their offspring. That's the name of the game. The animals that are alive on Earth are those whose forebears were able to propagate their genetic material by being well adapted to environments in which they had lots of babies or a few babies that they took very good care of. And the babies get to the point where they can acquire their own food and defend themselves or find a niche where they can avoid predation. And they need to be able to recognize the opposite sex, and they need to be able to attract the opposite sex, and they need to be able to mate, and they need to be able to make viable offspring. And then those offspring need to be able to get to reproductive maturity through survival and resistance to famine and resistance to predation and cleverness and so forth and so on. So I've often explained that, so it's a little bit of a head scratcher why any animal should live beyond their reproductive maturity. Some animals don't, right? You know, famously, there are some male insects that contribute sperm and then are like gobbled by the female because you've done your job and I just need that carbon from you. So thanks. And now you get into mammals, and we're mammals. Mammals are going to be closer to the human system than insects. Why is it that foxes can live sometimes 5 or 6 years? In the northern hemisphere, foxes are usually born in the spring, and then they can usually reproduce by the fall or the winter. Now there's babies within a year of a fox being born. But a clever fox may be able to reproduce 4 or 5 or 6 times as many times as a non-clever fox by being able to hunt better, by having more sex appeal, by protecting their babies better, and so forth. Longevity is the ability to keep your stuff together beyond reproductive maturity so that you can reproduce more times and take good care of the youngins so that they have a better chance for survival. So humans are at reproductive maturity by our early to mid-teens. And humans live into our 80s or 90s, right? In places that have decent healthcare and clean water and food, with reports of people living to 100, 110, and rarely 120. So is there a single longevity gene? No. There's going to be thousands of different genes, alleles of which have to do with how long the heart can function, how clever the brain is going to be, how to maintain skeletal muscle mass, how to maintain sex appeal, how to take care of your kids, how to avoid starvation. Unfortunately, some of these genes that involve how to avoid starvation, which allow us to turn carbohydrate into fat, they turn out to be bad for the current environment in which we have unlimited food available to us. So that's the big picture of the genetic control over longevity. Then you have people that choose their favorite thing that changes in aging, as you say, telomere lengths or DNA methylation, or they find or they observe correctly that when we do an in vitro induced pluripotency, we're taking a mature differentiated cell back to an undifferentiated state with Yamanaka factors, right? And we're reversing the life experience of a cell back to a pluripotent state in which it can differentiate into other cells. And then you asked me a series of questions about those. Are some of those hypotheses hyped and overblown? Totally. Would I invest in a company that wanted to increase telomerase activity for longevity? Never. Would I let any member of my family do that? No. If I were asked by a investment consulting company, which happens from time to evaluate the tech, I would say, we're talking hypothetically because I don't know what company you're talking about, but I would point out some of the same first things that you noted, that there are telomerase inhibitors for cancer. We immortalize cells with telomerase, and then we can make them procarcinogenic, et cetera, et cetera, et cetera. There are all sorts of problems with methylation clocks. Are you measuring a cause or effect or both? For sure. And so, yeah, it's a hazardous area. For me, I can embrace the concept of bioacceleration and, let's all come together and put the best minds and the best ideas and the best tech together and be transparent and put data available on the blockchain, and all of those things sound great. But then the foundational experiments and the foundational assumptions should be based on the most reproducible things in science, and not in an area that is notoriously hyped and is notoriously affected by publication bias, if you want me to.
Yeah, that's an interesting point that you say about putting research funding on chain, because usually when people hear that concept, the thing that they're most skeptical about is blockchain. But you're actually critical of the fundamental science that's being done rather than necessarily the mechanism of funding, or even maybe a little bit the type of the people investing. But it seems to me you're most critical of the actual science. Professor Brenner, you're one of the few scientists in the space who are willing to bluntly say that aging can't be stopped. And you're quite famous for criticizing the longevity hype, right? I guess my big question is, why aren't you bullish despite doing research that many would consider to be part of the longevity space? You're also an advisor to a number of companies that are like aging, longevity adjacent. It's interesting that you are very public about these opinions. A lot of people might say that this seems slightly misaligned, like the research and things like that. So yeah, maybe just what motivated you to really want to call this out?
So I was minding my own business working on an enzyme in 2003 for when I discovered the nicotinamide riboside kinase pathway. And I filed for IP for uses, both therapeutic, that means drug, and food and nutritional uses of nicotinamide riboside. Those patents were issued in the US and globally, and it created the NAD boosting industry. And as you said, I'm the chief advisor for Niagen Bioscience. It was formerly called Chromadex, that initially licensed the nicotinamide riboside IP. And yes, I advise other companies. I'm the chief scientific advisor of NADMed, which measures NAD with kits. They're based in Finland. I'm on a board of the Institute for Healthier Longevity, Abu Dhabi. There's a skincare company that puts nicotinamide riboside into products that I co-founded and help advise. But what you don't see any of these companies do is hype or claim that they can make you live longer. Because I don't think that there's anything in the human literature that suggests that will be achievable. Some people may think it's going to be achievable, but I don't think that they can provide any evidence for that. The use cases for nicotinamide riboside are that there are many conditions of metabolic stress in which the NAD system comes under attack. In many of those conditions, the nicotinamide riboside kinase genes, NRK1 and NRK2, get upregulated, and the cell or tissue is literally telling you that it needs or wants nicotinamide riboside to restore homeostasis. So if we confine our clinical trials to those kinds of spaces, right, where there's going to be an obvious metabolic use case for NR, we're going to achieve success. If I try to raise money, hundreds of millions of dollars, to do a clinical trial of lifespan extension for nicotinamide riboside, first of all, I don't think I could raise that money. Second, I don't think it would work. Third, it wouldn't work. And I don't want to waste other people's money. I don't like to waste my money, and I don't like to make promises that can't be delivered on. So I think that the more people ground themselves in achievable goals, the better they are. I think that some of the people that are investing in the space are investing in hopium, which is not a good foundational principle. And some of the stuff in the literature is really problematic because, and I do want to explain what publication bias is. So publication bias is when only positive results get submitted. So if I were to do a study that asked the question, can some drug extend lifespan in a mouse, or can some overexpression of some gene extend lifespan in a fly or a mouse, the positive result is exciting. The negative result is not, right? The principal investigator have asked a student to look at whether resveratrol extends lifespan in a mouse, and it doesn't. And so they don't publish that negative result, but literally dozens of research groups have that result. Dozens. And the papers, the most famous papers from Bauer and Sinclair from Harvard that attempted to show that resveratrol extends lifespan in a mouse did not show that resveratrol extended lifespan in a mouse. They had some other weird things in the paper and claimed that the overfed mice were more metabolically healthy, but they published it as though it were positive results. But if you were dispassionate about it and you said, no, my expectation was that it was going to increase SIRT1 activity and extend lifespan, and it didn't get that result, you should have published that as a negative result. Very few people do that. And so you end up with things in the literature that are not reproducible because one person gets what they think is a positive result, 9 or 19 others get a very non-remarkable result that don't even go into the literature. And then naive investors think that there's some new thing there and that there's some investable thesis. And a lot of times there isn't.
Quick question. Is it common for if someone publishes a positive result and a group of researchers find that that's maybe not true, either they can't replicate it or they realized it's a media effect, whatever. Maybe they can't, but can they critique the article, or there are mechanisms where you can respond, right?
It's incredibly hard to do that. So yeah, there are letters to the editor in some, not all journals. There's a category of kind of correspondence papers to journals that are called Matters Arising is to the editor, there's some serious problems here. But the editors have to agree to consider a letter to the editor or a Matters Arising. I've done this 2 or 3 times, and it takes an enormous amount of effort. I'm trying to direct my lab to do various things. And all of a sudden, we're reading a paper that appeared in the literature that we know enough about the methods to know that it's deeply flawed. I can think of 2 or 3 examples where I've gone to the trouble to do a Matters Arising or a letter to the editor. One was somebody claimed that there's an NMN transporter. They named a known sodium transporter, and they said this thing transports NMN. And they use terms in the paper that are not even scientifically valid terms. Like they said, NMN enters cells immediately. If you've taken any like high school or collegiate chemistry, things don't happen immediately. Everything requires time and temperature, right? So someone shouldn't have even had a paper reviewed if it said something occurs immediately. And then if you look at the way they prepared the samples, they are saying that this NMN entered cells and you'd have to wash the cells and see whether it's inside the cells or associated with cells. And they didn't do that. And so there were just a bunch of things wrong with the paper. When the journal agreed to consider this correspondence about a paper claiming that there's an NMN transporter, they also said, okay, we're going to allow the original authors to reply. So then they have their original paper, there's a reply from Schmidt and Brenner, and then there's a reply to our critique by the original authors, basically saying, oh, don't worry about it, it's all good. And it ends up just basically confusing readers. And I was an excluded reviewer, right? Because I developed the metabolomics technology. And I know that things don't happen immediately. So I'm excluded from review. And they have reviewers that may not be as critical as we are. And then they end up responding anyway. So there's a lot of obfuscated literature, is the way I would put it. There's a lot of things that should have been retracted. If you looked at Science magazine yesterday, or the New York Times yesterday, I think I'm referring to July 24th, 2025, because people are going to be watching this podcast in the future. Right around July 23rd or 24th of 2025, Science magazine finally retracted a paper claiming that there's an arsenic-based life form from Mono Lake here in Southern California. They said there's this really weird microbe that is growing in Mono Lake, and it doesn't have phosphate in its RNA or DNA. It has arsenate. It's an arsenate-based life form. And most biochemists and microbiologists were saying, did they really demonstrate that? No, they didn't demonstrate that 10 or more years ago when it was initially published. There was a lot of correspondence about it, and there was a failure to replicate it. The original authors did not want to withdraw the paper. They didn't want to retract it. But finally, the editors of Science said, look, we're the editors. We've seen enough here. We've seen failure to replicate. We can evaluate the nature of the critique and the nature of the defense of the paper, and we believe the critique more than the paper, and they retracted it. But that's very rare. And there are papers that I and colleagues figured out cannot be true and didn't prove any of the main points of the paper that we've asked major journals to retract, and they don't retract them.
That sounds kind of like the Tessier-Levine scandal, where the Stanford professor who didn't want to retract his articles, even though there was numerous people saying like these images are faked, or lots of different critiques, not even just about the fundamental science, but about data falsification. So this, I'm sure, is a very rampant problem that happens.
So data falsification makes this whole process easier. Okay. Or we've gotten a copy of the original spreadsheet, and when we calculate standard deviation and error and p-values and so forth, we don't get the graphs and conclusions that they get. That is an easier critique to make than the authors are selectively cherry picking images and putting together a narrative that is not supported by the data. Most of the problems in longevity papers have to do with cherry picking data and not replicating things well and not being blinded to treatment. So, for example, if you're claiming, if a principal investigator has already gone out and sold a book and said, we reversed a condition using these 3 Yamanaka factors, and they've basically already told millions of people about the results, and then, but they haven't submitted a paper yet. They've put their laboratory members into a very difficult situation because the lab's reputation is based upon the result coming out a certain way. There's quite a well-known case in which a paper was published in Nature in which the results were pre-announced in a book. And then when you look at the details of the methods of the paper, they say the investigators were not blinded to when they evaluated the mouse vision and the mouse visual system, which means the lab people are looking for a specific result from specific mice. We never do it that way in our lab. If we have a prediction, we don't want to be biased by that prediction. So if we think it's going to come out a certain way, that a certain mice are going to have worse fatty liver than other mice, we section them, we number them, and we give it to somebody else to evaluate it, right? And then we see whether we were right or wrong, right? We learn a lot of things when we're wrong. But there's so many different types of bias. So publication bias, as I described, and this is a kind of bias that comes from non-blinding, right? That's why, clinical trials are done with blinding, right? And preclinical work, the investigators' expectations and hopes should not enter into data analysis.
Yeah, and it does no one any good if you get a positive result in preclinical. It really serves no one in those cases because it does have to.
It's really hazardous for the field, in fact. So hype hurts, right? Because then you get investment in the wrong things. You get time and treasure spent on the wrong things. So that's why it's really important to have the foundational literature having been reproduced by other people.
Yeah, it hurts the field, the investors, and the scientists who are doing the work, and even the people you would expect to have the biggest financial incentive for doing it because they have to go for it, and they, yeah.
And certainly
Totally agree.
Yeah, I want to, so we've talked a lot about what you are most, I think, skeptical of when it comes to this movement. And I want to talk about it very broadly. I mean, we've been sort of touching on what the regulatory might look like for a longevity drug, for instance. And you mentioned at the beginning that, what would a clinical trial even look like for a longevity drug? Would we have to test for an entire generation, 2 generations? How long would these clinical trials be? I mean, it seems unfeasible, but there's so many companies out there with this claim that we have this one-size-fits-all drug, and aging isn't formally recognized as a disease indication by the FDA. I'm sure you've had some experience with this in your various companies and products. How would you craft a clinical trial that might actually be testable, or do you think that it's not actually possible?
Okay, so the biotech I should have mentioned is Alphina, where we are actually targeting the NAD system for particular types of malignancy, where we want to kill cancer cells, right, through inhibiting the NAD system in particular genotypes of cancer. So target selection, patient selection is going to be very important for the drugs that are developed in the NAD space, right? Otherwise, they would be generally toxic and they would just harm patients, right? Nobody goes into cancer therapeutics aiming to kill all cancers all the time. It makes no sense, right? You want to understand cancer genotype and targets and have a specific molecule for a specific indication. When you say this field, I'm not totally comfortable because I feel like I'm a biomedical scientist. I told you I can be on board with something called bioACC because that's what I do. I'm not sure that I'm on board with a path to testing longevity drugs in humans. That said, Loyal has an interesting approach, right? Where they say our companion animals, dogs, particularly big dogs, and you heard mine earlier in this podcast, they don't live that long. So we can do trials with dogs, right? And they can look at a whole bunch of biomarkers, but they can look at fitness, right? They can look at endurance and fitness and a lot of other things. I'm not an advisor, so I have no inside information on Loyal. But I know that the basic idea is to try to get approval for a drug in large dogs, right? Then see whether it will benefit all dogs, and then see whether some of the target stuff and what they've learned from their canine work can apply to humans. But I still think that when they do human clinical trials, when they or other sensible companies do human clinical trials, it's going to be age-related disease and conditions. It could be sarcopenia, right? It could be maintenance of skeletal muscle mass in people that have had cancer chemotherapy. That's a great indication, right? Now, you could get approval as a drug against sarcopenia, and potentially you could try to test it in larger populations of just aging people, right? And see whether it helps them maintain their skeletal muscle mass and function. But that's a discrete endpoint, right? I don't see, to me, if I were a regulator, I don't see the path to purely using biomarkers of aging as a regulatory goal or a primary endpoint of a clinical study. And none of us have enough time to do an actual longevity trial unless it's a progeric condition. So if it's a disease of accelerated aging, right, where the life expectancy is 25 or 30, and you enroll people who are 16, you may get a signal. But again, the primary endpoint might be their walking speed or their exercise tolerance. So I really think that when people soberly look at what the clinical trial path is, you need a primary endpoint that you can measure that relates to disease or some type of functional metric. And so I think the companies that succeed are going to be doing that.
Yeah. I mean, I think the general theme that I'm getting from this conversation is that we're a long way to go. Like, there's so much that we don't know about just the underpinnings of why we age, of many of the mechanisms we don't know, how NMN gets transported into cells.
I think I could explain how NMN gets into cells. NMN gets dephosphorylated to nicotinamide riboside and then goes through ENT1 and ENT2. So NMN doesn't get into cells by itself. NMN gets dephosphorylated extracellularly and goes into cells. But I didn't call NR or NMN longevity drugs. They're going to be used for specific disease and conditions in which the NAD system comes under attack. And I think it would be unreasonable to expect them to have an overall effect in lifespan extension. Now, that said, if you wanted me to try to defend the proposition that they might, I could say if NR is demonstrably anti-inflammatory, which it is, and if it can increase resistance to infection, then over time, People that are boosting their NAD with NR may be more resistant to infections, and over time, infections and inflammation can kill you. So yeah, there could be a way to get a signal there, but to me, that's not a way to run a clinical trial. You can run a clinical trial for resistance to flu during flu season, and you can get a demonstrable effect in a few months. That's the way people are going to run trials. People are not going to run trials that run for 5 years unless they absolutely have to.
Yeah, it's hard enough already, so I can't imagine a 50-year-long clinical trial. It just seems absurd. I just had one kind of conclusory question that I think will help wrap this conversation up quite nicely. What is something genuine that you are excited about here? I mean, I think that, you are a biochemist by training, right? And that is what your lab focuses on. And you never expected the NAD hype to become associated with aging, I think, or that was clearly not your goal. NAD, as we've discussed throughout this entire conversation, is an extremely versatile pathway, and it's involved in so many other different processes in the body. And so this was clearly not your, like, I want to become the number one aging guy. Like, that is clearly not your thing. Is there anything that anyone is doing out there that you're like, this could actually work?
So the thing that the Brenner Lab is really excited about right now is what we've learned from a rare disease called citrin deficiency, CITRIN. So this is possibly one of the most common rare diseases that you've never heard of. It may be as common as cystic fibrosis, CF, and it's caused by a mutation of a gene called SLC25A13. Most of the global disease burden is in the Far East, and it inactivates something that is well known to people that work on NAD, namely the what's called the NADH shuttle system. It's really nerdy. It moves 2 electrons from the cytosol of a hepatocyte into the mitochondrial matrix by transporting glutamate and aspartate. It's an amazing system. But it's a very fascinating disease because it is right at the crossroads between carbohydrate metabolism, protein metabolism, and lipid metabolism. And it disturbs all three of them. And the kids that with citrin deficiency have a strange sense of taste. They don't like sweets. They're very lean, and they developed fatty liver disease. And we have a new mouse model or existing mouse model of this disease in which we figured out a lot of the hidden puzzles of citrin deficiency. We figured out that the fatty liver disease arises from activation of a transcription factor called ChREBP. We found an unanticipated activator of that transcription factor. We figured out why people with the disease don't like sweets, that they circulate a hormone out of their liver called FGF21. We have an idea of how to use the new knowledge that we've discovered to create drugs that will induce FGF21. And we have some new ideas of how to target fatty liver disease as well as urea cycle disorders by the work that we've done. So we're incredibly excited by the work in this space, and we think it has the potential to produce therapeutics for fatty liver disease as well as sweet tooth and even alcohol use disorder. So that's what excites us the most these days.